We are very much looking forward to your talk, Marinka. Welcome on stage.
Thank you so much for inviting me. I'm excited to be here, and thank you for this nice introduction and the anecdote as well. So today I will talk about machine learning for drug repurposing, and I will also try to really build bridges between kind of machine learning community, which would be my community that is my home in research, and also visualizations and area of research where I see more and more really critical gaps are there that need to be filled, and hopefully this talk will also create some opportunities for discussions on how to close these gaps and kind of leverage the opportunities that are there. So the focus will be on drug repurposing or drug development. So let me just very briefly talk about that. It has been addressed in a number of talks at the conference already, but very briefly for the purpose of today's talk, we think of drug development being a complex, multi-year process that consists of a number of steps, and those steps act as a funnel. So increasingly we go through a large number of compounds that might be that represent candidate drugs initially, and through a series of steps then we finally reach, after on average 12 to 16 years and spending $1 billion to $2 billion, to a new drug that would be then approved by FDA input on the market. There are a number of opportunities for machine learning in each and every of those 5 steps that drug development consists of. So in step 1, which would be that of drug design and discovery, there are opportunities for machine learning methods to do synthesis prediction, think about models that can do de novo drug design, that can predict properties of molecules, and those properties that might be of interest down the line in downstream experiments and development for a particular disease. In second step, in preclinical research, then methods exist that can help drug developers with virtual drug screening, drug target identification, answering various questions about safety and animal testing. Then going to clinical research, certainly an important question relates to clinical trial recruitment, predicting whether drugs can be safe and effective really on humans. And then in step 4, which is that on the FDA side, reviewing the documentation as well as putting the proposed drug in larger context before the approval, and finally step 5, after the drug has been approved, then opportunities for machine learning are certainly abundant, especially related to post-marketing and surveillance and safety monitoring of drugs that are already on the market. So this is really exciting for someone who works in machine learning, because now we have all these amazing opportunities, and more data sets are also being generated that can allow us to start tackling those questions computationally. Now, it is challenging to realize this vision. So what prevents us to really capitalize on all these opportunities? For the purpose of these talks, I will primarily focus on 2 main challenges. So the first challenge shown on the left here on the slide is that finding what drugs work for diseases, what drugs will treat disease, really relies on complex interconnected biology. When I say that, what I mean is that it is not enough to simply know what are the proteins that the drug will bind to, but it's important to know how those effects on cells, on proteins that the drug has, how those effects propagate through underlying biological networks, and how they go to this multi-scale view of the human organism, and then in the way that they would eventually manifest through clinical the clinical patient level. So there is this question of how we can really tame this complexity of the data and the problems that we have. And second major question that kind of prevents us from realizing this vision of how machine learning can really assist and expedite drug development is really that what we need are not only predictions that are accurate, but we need predictions that I will call them in this talk actionable. When I say actionable predictions, I mean those are predictions that of course they need to be accurate, but that's not enough. They need to be robust, interpretable, and also detailed or precise or very concrete concrete, so they are really useful to domain experts who can then take those predictions and run experiments in the wet lab, or thinking about what are the might be most promising candidates for further investigation in the lab. So in today's talk, I will address and talk about some of the progress that we made on those two challenges from 3 points in the context of 3 themes. In 3 topics. So first, I will talk about our approach for expanding the use of drugs for new diseases. So this is the drug repurposing problem, where I will outline some of the machine learning methodology, but then also talk about how we can integrate that to enable feedbacks for the AI loops, make predictions for never before seen diseases, and think about doing repurposing at the pandemic scale. Then I will talk about in the second part on how we can use knowledge graphs to answer complex logical queries to make predictions really actionable and useful to domain experts. And finally, I will conclude by outlining some of the ideas and progress that we made on modeling safety and side effects of drug combinations. So let me start with the first part, which is that on drug repurposing. So when we say drug repurposing, so what we mean is that it's an alternative appealing strategy to drug development, where we would want to develop, where instead of developing a drug from scratch, we would look at what are drugs that are already on the market or that are already in late stage of development, or potentially were on the market but were removed from the market for some reason. Do any of those drugs potentially would be useful for treating a particular disease of interest? So in a way, what we are looking for, we are looking to find new tricks for all drugs. We try to find how to repurpose, extend the use of current drugs and use them for diseases that we care about. In particular, this is an appealing strategy. Why? Because drug repurposing tend to operate on a much shorter time scale than development of a new drug from scratch. As well, in general, it is much less expensive than developing a new drug from scratch. So there is certainly quite substantial interest from the academic as well as in industry side on how we can identify what drugs could actually be useful for treating the disease. So computationally, the way we think of this problem at the very high level is that we think of the problem as having a large universe of drugs, which I'm showing here in a schematic on the left side, and having a large universe of diseases on the right side. And we know already for those drugs what diseases they are indicated for. We know what are diseases that those drugs use. So our goal here will be to establish these additional relationships between drugs and diseases in a way that those relationships would be indicative of what new diseases a particular molecule or a drug could treat. So our key insight to tackling this particular problem is that we will really want to represent the complexity of the interconnected biology through networks. So in this particular slide, I'm showing one network or a graph which will be our schematic for a protein interaction network. So this will be a network where every node, here white circle, represents a human protein, and two proteins are connected if they physically interact with each other. So what we do in this is take the human PPI network and we overlay it with a number of drugs and diseases from our universe of drugs and diseases. So here, just one example would be a drug disease on the left, a part of the slide, which would be a subgraph, a subnetwork of the entire protein interaction network, represented by or defined on a set of proteins that are associated with that disease. And on the other hand, drugs on the blue subgraph on the right is just one example of a drug represented by a rich protein subnetwork defined on proteins that are targeted by that drug. So in pharmacological sciences, there are many notions that are used to characterize and try to model the situations when a drug would be useful and have some desired therapeutic effect on the disease. And often it is said that a drug will likely treat the disease if it's close to the disease in some pharmacological space. Now, what does that mean? It really means that this pharmacological space is abstract space, which is hard to computationally operationalize. Our idea here is that we use the power of embeddings, which I will define in the next slide, to operationalize this concept of what does it mean that a drug is close to the disease in pharmacological space, to make this concept computable such that we can learn and reason over it and then make predictions about drug-disease relationships. So really the key concept here is that of embeddings, and this technical notion refers to the situation where we are trying to represent a particular object of interest. In our case, those would be drugs and disease subgraphs with vectors of numbers in some compact low-dimensional space. So more concretely, so in our case, we would start on the left with our protein network on which we overlay many drugs and many diseases. Here's just an example with one disease, adenocarcinoma, and one drug subgraph, erlotinib. And then our embedding function, here denoted by F, is a function that maps each and every subgraph from the left to a point in that low-dimensional compact space in the right, such that two subgraphs on the left that have similar topology or are proximal in the underlying protein interaction network would be embedded close together in the space. So in doing so, we can really operationalize this concept of being close in the pharmacological space, and we can make it computable. So really the question becomes what's hiding behind that function F that we are outlining here on the slide. And the goal of that function F is to really map subgraphs to this d-dimensional embedding space so that those subgraphs that are proximal in the network, that have similar topology and are similar in other aspects of graph structure, they would be embedded close together as points in that embedding space. Imagine once we have those embeddings, what we want to do then, well, what we want to do next is we want to combine embeddings for drugs and diseases in such a way that we can predict new relationships between drugs and diseases. So really a prediction problem, prediction test is that given a drug C and disease D, we want to predict if C will treat D. And the way we think of it is we start on the left, which is universe of drugs and disease subgraphs, our method, which we call sugar learn embeddings for those drugs and diseases. So for every drug and disease, we have this low dimensional vector representing the drug and the disease well, and summarizing the drug structure in the underlying biological network. And then the method combines an embedding of a particular drug and disease and comes up with a score representing how likely a drug will treat the disease. So the core idea here technically is that of how do we really specify that mapping function F that will generate our embeddings for drugs and diseases. So how do we learn those embeddings? And here really we capitalize a lot on advancement in a relatively recent young field of graph representation learning on graph neural networks. Intuitively, the way you can think of it is that we think of every drug subgraph and every disease subgraph, here this blue example would be drug C, as a kind of super virtual node or virtual object that is able to propagate messages, those neural messages from that virtual object representing drug to the underlying protein interaction network, so to the proteins that the drug binds to, but then those messages are also shared, propagated within the PPI network, and then eventually aggregated and sent back to the drug node. Now, all these steps are learnable, meaning they are being optimized during the process of training in such a way that the resulting embeddings that we obtain by propagating those messages from drugs to PPI networks and then back give us the best possible embeddings that can capture the topology well. And this is done through this two-step process that consists of the encoding stage, where we generate embeddings for drugs and diseases, and in the decoding stage, where we take those embeddings as input and we decode them into predictions representing probabilities that a particular drug will have the desired effect on the disease. So when we have developed this approach, the first thing we want to do is really understand how well does that framework and technology work on data sets and drugs for which we already have some treatments. So in order to be able to do that, we first spent quite a bit of time constructing a drug repurposing data set that we also shared with the community, that consists of a number of data sets from various sources of biological knowledge, including protein interaction network, which was this underlying network represented by the white circles here, as well as many drug disease subgraphs obtained based on drug target information, based on disease associated proteins, and lots of side informations. What does that mean for this? The side information are additional attributes or feature vectors that represent and encode more information about drugs, their molecular structure, more information about proteins, their gene expression, the gene expression profiles of the encoding genes, as well as more information about diseases and symptoms and so on. So this gives us now this rich network where we can think of it as the underlying knowledge graph, or which is highly enriched protein interaction network with lots of subgraphs on top of it. Once we constructed this data set and trained our model Sugar, we first compare it to a number of existing methods for drug repurposing. And I will go fairly quickly through that. What we noticed is that a couple of things. The first thing I want to mention is that as many others, we have found that particular representation learning embedding-based methodology leads to really substantial gains in performance over some alternative methods, which are also very powerful, that use random walks or other types of machine learning models, such as SVMs. And we were also very excited to see that by using embeddings and training them and learning those embeddings in the most optimal way, we can actually capture this notion of closeness in pharmacological space much more accurately than, for example, simply calculating various network science metrics, for example, those based on shortest path lengths or some other means, such as network kernels and so on. So that was really exciting to see. And then we started to ask, well, now that we know, or like we have convinced ourselves to this experiment and many others, that the method performs really robustly, that it can provide accurate predictions, we want to see how well can really how well can it perform in practice. So we teamed up with Stanford's Spark Translational Research Program, and we asked that program to share with us some of the drug disease pairs that and some of the list of drugs that were repurposed over the last a decade or so within this Spark International Program and Center at Stanford. So this is just part of the list here, but on this slide, the way you can read it is that it has three columns. The first is a list. The first column is a column of drugs. The second is a column of diseases. And then we know that, for example, carvedilol was repurposed for Chagas disease. And we wanted to ask our model, how highly would the model predict carvedilol, or how highly would the model rank carvedilol to treat Chagas disease among all other possible drugs that we have in our system. So this is what the data in the third column represents. The way we read that is, for example, for the case of carvedilol, it was ranked as number 9 drug out of 5,000 drugs that we had at that point in the system, meaning that this was, according to the model, 9th most likely drug to be useful for Chagas disease. As you can see also for other examples, those drug ranks are relatively small numbers, meaning that the right drugs or those positive hits were actually ranked by the model very close to the top, which is a very encouraging result to us. And that has led to a number of follow-up research where we are thinking about integrating and like working on models that can make predictions about drug disease treatments in the context of prostate cancer, where in specific the question is how to make personalized predictions that would make personal level treatment recommendations for a patient, and also would minimize certain undesired side effects. In that example, that would be incontinence in the context of prostate cancer. So recently, we started in March actually, we started to ask ourselves, well, how can we deploy this methodology to be able to contribute into discovery and of therapeutics in the context of COVID-19 pandemic? And really, how can we deploy this technology in order to be able to expedite and support the development of effective and safe medicines for COVID-19? So in particular, because of the disruptive nature of COVID-19 pandemic, it seems very hard to be able to expedite the process of drug development in the context of developing a molecule from scratch, to expedite it so much that it would be possible to get a new drug developed completely from scratch in such a short period of time. And because of that, this exactly creates opportunities for drug repurposing. So it's a more realistic strategy here, because it allows us to really be able to come up with a list of drug candidates fairly quickly, and some of those might already be on the market, so or in a way in late stages of clinical research, and therefore we might require shorter, less time to put those drugs to patients. Why is this really challenging from the machine learning standpoint? Working on drug repurposing for COVID is really challenging because COVID is an example of never before seen diseases. So why I'm mentioning that? Because often when we train our machine learning models, we require some information about successful examples, so examples of, for a given disease, or examples of drugs that are already used for that disease, and we would want to expand that set of drugs by pulling in new drugs. And that, of course, does not work for COVID-19 since initially, back in March, very little data was available, and still this seemed to be the case. So in the context of COVID-19, we used the methodology that I briefly outlined before, and we applied it to COVID-19 disease. And the first thing what we really need to have in order to be able to do that, we need to have a disease subgraph or disease module representation of COVID-19. And here we really relied on the work that Gordon et al. From UCSF have done and found a set of human proteins that the virus binds to, and those set of proteins were used by us to define COVID-19 disease module, which is then now a subgraph shown here on this schematic as a viral disease module. It's a subgraph of protein interaction network that is defined on those COVID-19 associated genes. And then once we have this representation for COVID-19, the approach that I outlined before allows us to rank all drugs that are currently on the market with respect to how likely they are to be effective for COVID-19. And we can do that even though there are no existing COVID-19 treatments available out there. So the approach that we used to do that was consisted of a number of different models. And here I'm just outlining it at a very high level. So we considered approaches that are based on characterizing proximity or closeness between drug and disease subgraphs, so between COVID-19 module and subgraphs of many other diseases. by characterizing those proximity through various network medicine metrics. And we also considered methods based on random walks or diffusions, and also then a number of, and then also methodology based on graph neural networks, which is the method that I have outlined before. And all these different methods have produced a predictions that we then combined in order to come up with a list of ranked drugs that or prioritized list of drugs for COVID-19. And coming up with that list, we wanted to understand, well, how does COVID-19 relate to other relate to drugs, relate to diseases? How does that pharmacological space that we talked about, what is the structure of that pharmacological space in the case of COVID-19? So I'm showing that pharmacological space as instantiated through COVID-19 and embeddings of other drugs and diseases. This is shown on the left. And you can see that once we have the embedding for COVID-19, we can look, well, what are drugs, other drugs that are embedded close together to COVID-19 in that space. And those drugs would then be some of our most likely candidates for repurposing. In doing so, we of course looked at what is the performance of or predictive ability of different pipelines that we have in our models. And here it's really, while that might seem a very standard way to be calculating these performance metrics and performance curves, of course, the main challenge is that there is very little ground truth label information on that. And here because of that, we used clinical trial data and a set of clinical trials on COVID-19 as a very rough proxy that give us, to obtain some insight whether our approach and the individual pipelines that we have here based on graph neural networks, based on diffusion pipelines, D1 to D5, and based on proximity metrics, whether they are able to construct, make predictions that put at the top of our ranked list those drugs that a research community thinks are currently promising or worth exploring in the context of clinical trials. But then what we really wanted to do is, well, once we realized that, well, we are able to perform remarkably well in the context of identifying what drugs will be studied in clinical trials for COVID-19, we wanted to see, well, how useful would our predictions be for really experimental work. So because to do that, we teamed up with collaborators at BU and the National Emerging Infectious Disease Laboratories that Boston University has, where they can do experiments with dangerous pathogens. And we shared with them a prioritized list of our predictions. So this is the list shown on the right part of the slide. So the way this list looked like, it has essentially two columns. The first column was a list, were name of the drugs, and then was the ranking of the drugs according to outcomes predicted by the pipelines. So these were the two key columns. Of course, we provided also list of drug targets and which biological functions and so on, but that was the key prioritized list that we shared with them. And those that might be interested in follow-ups in that, I encourage them to read our draft that is available on archive, and we will have a follow-up very soon in the next week or two, sharing the results of experimental screening, which are very promising. But what I really want to talk about here in the context of BioVis is this question that I got while working on this project, as well as other projects and collaborations with domain experts. And the question was, well, how can we go beyond this static prioritized list of repurposing candidates? Beyond those lists, for example, as I showed you on the left, on the previous slide. How can we make predictions that are really actionable, meaning not only accurate, which we have some handle of how to make predictions really accurate with state-of-the-art models, but also predictions that are interpretable, that are detailed, that in a way are interactive and allow domain experts to interact with our models. Or in other words, how can we repurpose drugs at the pandemic scale? So to be able to really deploy our models very, very quickly and kind of make this progress such that it can be, you make this a strategy such that it can really be used and deployed at the pandemic scale. So the way I've been thinking about that is that, well, we started developing a methodology that can then introduce feedbacks for the AI loop. And I'm highlighting this entire pipeline here in this slide, where the idea is that we have domain experts that ask those different kinds of queries or questions. For example, would benzamyl treat psoriasis might be one simple question the domain expert would ask. And then the underlying model based on protein interaction networks and those drugs and drug and disease subgraphs that we have seen earlier in the predictions would generate embeddings, produce predictions, and present those predictions to domain experts that can then ask questions such as, well, what data could explain those predictions? What if I would change the data a bit? How would that affect my predictions? Or provide feedback for the AI model in the sense the domain expert can say that certain predictions make more sense or make less sense, and then the model is able to adapt to that feedback. So really what is the core missing part here is that allows domain experts to understand, and get better view into predictions made by the model that can ask what if questions rather than just get static list of prioritized drugs. So in that line of work, which we started focusing on the problems of how to explain predictions made by state-of-the-art models for machine learning on graphs. And the key idea that we are trying to, that we are modeling here is that we want to summarize where in our large data set, where in our rich protein interaction network, the model looks for evidence for a particular prediction that the model made. So we want to find what is the part of our entire data set that's most influential for the prediction, the part that drives the prediction made by the model. So formally, this is done by using counterfactual reasoning. And we published recently a paper at NeurIPS in 2019 of a model that can answer exactly this counterfactual questions that is called GNNExplainer, and we also deployed it in the context of COVID-19, and it turned out to be quite useful to domain experts to try to make sense out of predictions made by the model and then decide which are those that are most exciting to take to experiments. So, and the key idea of GNNExplainer is that imagine we have a particular prediction or particular input that we want to make prediction for. So that input would be a drug in the COVID-19, and our model F makes a prediction F sub X that would indicate the probability that the drug would be useful for the disease. So if that is the input, what the GNNExplainer produces, it produces an explanation, which was a small subgraph together with the set of node features. So these are all these side information that is encoded in the data, such as set of biological pathways on proteins, or molecular structure, or side effects of drugs, such that those explanations really encompass the critical part of the data that the predictive model F used to make the prediction. Meaning that if any part of that data is removed from the model, all of a sudden the model would not make that prediction at all. So we can find those critical parts very, very efficiently in linear time in their size with respect, or in contrast to doing that in a naive way, which would take exponential time in the size of those explanations. And we can then use those explanations to interact with domain experts and visualize and present those explanations and subgraphs to them and kind of get feedback from domain experts. So for example, what are some of the questions that one can ask? In one particular study, we used our graph neural network methodology to be able to predict properties of molecules, and that allowed then to domain experts to ask questions such as, why did you, meaning the model F, predict that the input molecule will have a mutagenic effect on gram-negative bacteria? And explanation would come in the form of parts of the molecular structure that is indicative. According to the model, that would be the reason why the model made that particular prediction. And then that allows domain experts to see, in some way, get a better understanding what's the part of the data that drives the model's prediction, how can we debug those models and then also make them better. Another example is just domain experts asking whether a particular drug, a form of statin, will treat hyperlipoproteinemia, and then domain experts could ask, well, why did the model predict that this statin will treat hyperlipoproteinemia? What is the disease treatment mechanism that the model has used to be able to make that prediction? And what the model would return, it would really highlight the parts of the entire data that the model has used to make that prediction. And that can then be visualized as in this small subgraph of proteins associated functional gene ontology pathways, and that explain what really has driven the model, to make the particular prediction. So this concludes this first part of the talk, which was the longest. So I promise that the two other will be much shorter, on how we can expand the use of drugs for new diseases. So how can we really do effective drug repurposing? And really my takeaway message from this part would be that in order to really make actionable predictions and to be able to deploy the drug repurposing or machine learning methodology at a pandemic scale, what we need are really powerful interfaces that allow domain experts to interact with machine learning models. And GNNExplainer, through the form of those feedback loops for the AI system, was one way that allow us to really make a step towards this goal. In the second part, I will talk more about the learning and reasoning over knowledge graphs and how that can be useful to further increase actionability of our predictions. So when we talk about knowledge graphs, what are those? So knowledge graphs are often called multimodal networks or heterogeneous networks, are networks or graphs when we have nodes or vertices of many different types of edges. So we might have many different types of that might be, for example, drugs, diseases, as we have seen before, proteins, metabolites. There might be single nucleotide variations. There might be other pathways or biological processes. And this complex data, when it's brought together in order to integrate and come up with one machine readable unified representation, this representation is then called knowledge graph. Now, once we have knowledge graphs, well, the reason why we construct them often in machine learning is because we want to leverage and use them to make predictions, to reason and learn about those knowledge graphs. In our case, we want to learn and use knowledge graphs that bring together very many different types of biomedically relevant data in order to be able to make predictions about safe and effective medicines. So far, the kind of predictions I talked about were those that were more or less very simple link predictions. So remember, before we were basically asking, can we predict drugs that treat diseases? So we had those drug subgraphs and disease subgraphs, and our predictions were simple edge predictions. We were predicting links between drugs and diseases. So what we argue is that, well, that is great, but it's not so actionable in the sense that when we really want to deploy these models, we don't want to simply say a drug will treat the disease, but really how will that be achieved, or how will that kind of, what are the follow-up experiments the domain expert can do in the lab to kind of get insight into that prediction. So that means that our models need to be able to answer more complex logical queries. For example, such a query would be predict drugs that target proteins, which in turn are associated with a particular disease D1 or D2 of interest. So this is much more complex logical query that involves multiple entities in our nodes in our knowledge graph than a simple two nodes and a simple edge between the two of them. So that means that what we want to do, we want to move beyond simple edge or link prediction to more complex logical queries because they are more precise and detailed and therefore useful for domain experts. So for that, what we have developed is a powerful toolbox that allow us to be able to very effectively answer any type of logical query that can be formulated in a class of queries known as conjunctive logical queries, which is a sub-part of the first order logic. But in very simple terms per here and at the high level, that means that we can effectively take a knowledge graph, learn a representation for or embedding for every node in that knowledge graph, and then also learn what is the best representation or embedding of logical operators. So what is the representation of logical AND operation? And once we have that, then we can effectively answer queries by simply combining embeddings of nodes on the knowledge graph, embeddings of objects such as drugs and diseases, with embeddings that represent logical operators such as logical AND and logical OR. And that can then handle many conjunctive queries, and we can do this really efficiently, so that we can then ask various types of queries on reasonably large graphs. And here's just one particular example, which is actually not so large, but it just used to demonstrate the utility of that approach, where we had a knowledge graph of around 8 million edges encompassing 5 different types of entities: proteins, diseases, drugs, side effects, and biological processes, and then 42 different types of edges between those entities. And we can then ask queries. We can then allow and enable domain experts to ask queries, and we can provide answers such as domain expert can predict what drugs will likely target proteins that are involved with diseases X and Y, where X and Y would be 2 concrete diseases that are of interest to domain expert. Or predict what drugs might treat diseases that are so in turn associated with a particular protein. And doing first classic cross-validation type of analysis, we can really show that this approach performs very well on answering complex logical queries, which would be this part particularly on the right, and at the same time it can also answer very accurately those classic link prediction types of queries. And here in particular, what I wanted to mention is that in doing so, we made a number of observations that have then spurred exciting new areas of research. One of them particularly being that many methods that are current state-of-the-art machine learning methods, primarily for graphs, that are being used quite extensively also in biological networks, are really founded on ideas that are based on other non-biological networks, such as ideas based on social information networks or social relationships. And often we find that those principles do not necessarily hold in the context of biological network. So here I'm just showing one example which is known in social information sciences as the friendship principle, meaning that if we want to know what a particular node or a person would like to do, then we look at its immediate neighbors or friends, and then we will say whatever sport activity friends are enjoying, that is also the sport activity that that person will enjoy. So if we translate this idea very naively to biological data, then it really does not necessarily apply. For example, if you want to predict properties of proteins, it's not necessarily the case that those proteins with which this gray protein directly physically interacts have exactly the same phenotypes as this gray protein in question, but actually we might want to look at proteins that are multiple hops away. Those kinds of observations that we came up are very much often the side effect of being able to answer more complex, ask and answer complex logical queries, so being able to probe the models, see what kind of data they're using for making predictions, and if that makes sense. And what is then now is that if you can encode these principles directly in our graph neural networks, in this case, we can very easily get substantial improvement in performance because we are encoding these biological principles directly rather than relying on some principles that were that really would not apply to the context of biology. And here's just an example where this principle of mutual interactors was applied to the problem of identifying drug targets and to the problem of identifying disease proteins, where we looked at can we label proteins based on their diseases that they are associated with? And using this cross-validation setup, we found that, again, encoding this principle directly in the model leads to substantial improvement over simply relying on using classical models without, that are really not necessarily, that are not well-founded in the context of biological, the nature of biological interactions.
So Marinka, just a slight second for me, please. Some people are wondering if we have enough time for questions and discuss. A lot of comments are sort of coming up in the Q&A. So I was wondering if you could, I hate to interrupt, but maybe come to.
Yes, I'm actually, I'm kind of at the wrapping phase. So
thank you.
To recap this part, we talked about the reasoning about safe and effective medicines in the context of knowledge graphs. And the two key messages are that knowledge graphs are really a unified representation that allow us to combine biomedical data in the broadest sense. And we can reduce under redundancy uncertainty in doing so. And then really to be able to make actionable predictions, it's important to go beyond classic predictions of edges. So to summarize the talk and leave more time for questions, in today's talk, I talked about several different problems related to different stages of drug development and drug repurposing in particular. So what we looked what we discussed was the application of applications of graph neural networks and machine learning methods on graphs for problems in virtual drug screening, drug target identifications, answering questions about safety in animal testing, then also predicting whether drugs would be safe and effective to test on people, and kind of another topic which perhaps can be left for some of the questions to think about how can we use this methodology to do post-marketing and safety monitoring. And really my final slide is here. Machine learning for drug repurposing, I think, creates is a really nice new toolbox that we can now bring to the process of drug development in order to expedite this analysis. On the other hand, there are a number of gaps that we need to fill in order to be able to expedite this process and deploy this methodology in real world, which was clearly seen during last few months, where we see how although very exciting, there are still many things that need to be done to be able to do repurposing at the pandemic scale. So I hope I outlined some of the opportunities as well as initial steps that we made toward this progress. And finally, all papers, code and data sets that I've been referring to, we are on our website and are available for everyone to use and download and kind of use in your studies. Thank you very much.
Thank you very much, Marinka. That was an amazing survey of your work, and I having a hard time following all the questions now that people are so excited about your work and have posted a number of questions. But let me maybe start with the first part of your talk, and a number of questions were posed with respect to the actual PPI network. In particular, so I mean, how resilient is sort of your method with respect to the embeddings? So meaning if you took your PPI, and are you sure about the completeness, or how much does it matter if the PPI is complete, or how would you interpret missing edges? Is it, you know, the data is not there yet, there has not been seen, or does it really not exist, right? And so that is one of the summary of many questions in that context.
So this is a great set of questions, and those that paid attention to what I refer to as actionable predictions were also predictions that are robust, meaning robust to incomplete data, missing data. So we have recently made quite a bit of progress on that with an approach I was not able to kind of discuss here, but it is called GNN-GART, and we have a preprint on arXiv. So what has been observed by us and several other groups is that when we train these highly nonlinear black box models on protein interaction networks or other types of networks that are generated experimentally, and then they are often incomplete because certain interactions were not measured, or because of limitations of the current biotechnology, certain edges might be there, but are not are actually not correct, so they are false positives. Many of these black box models are extremely sensitive to small perturbations in the underlying networks. So this is of course a problem, especially in the context of biological networks where the data is so complete. So the mechanism that we're using that allows us to think about this problem is that of adversarial perturbations, where we have designed now graph neural networks or models that we can add onto graph neural networks that can make each popular graph neural network model much more robust to small perturbations of the data. So it's exactly addressing the questions that were asked. It's called GNN-GART, and the preprint is on arXiv.
Yeah, so in this context, there is a number of further questions, and it would be great if you could also have a look at them during the break, or maybe we can shift over to the Cafe Connect. Some people were referring to also their, I see, also own work in that context about the PPI networks, etc. Some people ask about where did you get the databases or the data for the used PPI, as well as the disease protein interaction, etc., etc. I would ask, because we have also many other questions about your second part of the talk, to maybe postpone that to a later chat. So let me go to the obviously people are very interested in these COVID-19 predictions, and so some people ask, for example, for that one, I also noted that you Carvedilol, you said it was ranked number 9 under 5,000, and obviously the first question that comes to mind is, what were the first, I mean, the rank 1 disease or
the first 8 drugs, right?
Or the first 8, exactly. And
yes.
How much sense do these make?
Yes, so these are great questions. In particular, for the problem of carvedilol, as well as for COVID-19, those are really projects where we want to go beyond just computational validation or doing kind of in silico cross-validation type of studies. So what that means concretely is that we have collaborators on the experimental side that we work with, and they are validating our predictions in the lab in the context of COVID-19 that is on human and monkey cells, in the context of carvedilol that is currently using patient data from hospitals from the West Coast. And actually in doing so, as a machine learning person, I've identified or observed a number of these challenges that really we need to solve in order to be able to interface with domain experts in more seeming way, rather than just sending back and forth those tables with prioritized drugs. And that has really then, I think this is really where lots of exciting opportunities lie at the intersection of machine learning and visualization community, to be able to really deploy these models and go beyond just simply calculating in silico AUROC scores and precision recall curves.
In fact, somebody also asked whether you have contact or you're collaborating with pharma companies or hospitals for the practical use of your repurposing predictions. So that was actually answered already. Robin Kozierok asked whether the drug repurposing ranks is it per disease? So is it 11 of 5,000 predictions for a given disease, or are they overall ranks for any drug disease pair?
Oh, so that's a great question. So the rankings are per disease, right? Because if you would just make the overall rankings, they're like very hard to make any use of those overall rankings. What would even mean this drug to be useful for all diseases out there, or just for none of the diseases? So the rankings are always disease specific, and sometimes we go beyond that in the context of COVID-19, we are now making tissue-specific predictions. So which would mean that we would provide separate prediction lists for COVID-19 for lung tissue and separate prediction list for COVID-19 for brain tissue. And we would look at, well, what are the differences? What are drugs that we see at the top in one list and not at the top in the other list? And are those drugs, do they have targets that are tissue-specific targets, and so on?
Yeah, okay, that's what I thought. Actually, German Novakovsky asked, can we use this approach to identify drugs that can cause some desirable phenotype? So in the other direction, so to speak, right?
So that's a great question as well. So some work that we have done recently is related to modeling more directly safety, in the sense of predicting unwanted side effects of drugs as well as drug combinations. And here we can take this reverse approach, or where we often think of side effect as an unwanted effect that we want to get rid of. But while something can be unwanted side effect for one particular patient, it might actually be a desired effect for a patient in another situation. So we are doing exactly that in one project, where we are thinking about, well, how can we come up with a cocktail of drugs where each might have just one part of the desired effect, but altogether they achieve what we want, or perhaps a side effect of one drug is actually exactly what is missing from the current treatment that we have for some other disease.
Yeah, so we actually have come to an end, but maybe I can end this amazing list of really interesting questions. So you will be also, I think, very fascinated by the many questions, and also, I think, very valuable questions. And I hate to interrupt this, but maybe can we actually stop with a very quick answer to the question, how do you evaluate the actionability or actionableness of a prediction? I also found that a bit hard to quantify or whatever, right?
That is a great question. So far, we really don't have a way to rigorously evaluate actionability overall. So right now at this stage, actionability for us is a more abstract concept or umbrella term, under which we put up a number of desired properties that machine learning models would need to have in order to maximize their utility for domain experts, which were users of those models. And we can characterize each and every of those components, but overall the actionability it's hard to characterize. But what we can do, we can look at accuracy independently, robustness independently. There are methods to measure explainability or interpretable power to user case studies and so on.